Hello students, in today's class we'll study about artificial vegetative propagation. It is a method in which a complete new plant develops from the cut parts of the plant such as roots, stems, leaf, etc. It is that method which helps us to get a hybrid variety containing the character of both the parents. This is done by taking the rooted region of one of the plant and the shoot region of another plant and then combining the cambium of both of them and attaining a new hybrid variety. This method is practiced by 4 techniques by cutting, layering, grafting and tissue culture. The technique of vegetative propagation practiced on field are cutting, layering and grafting, while that practiced in laboratory is tissue culture or micropropagation. We start with the first technique of cutting. In this technique, we will take a small part of the shoot of the parent plant of around 10 to 15 centimeter, provided it contains nodes and internodes, and then we'll plant it in the soil. After planting in the soil, we'll observe that the adventitious roots start developing from the lower node. This type of method is practiced in plants such as sugarcane, roses, grapes, and bougainvillea. Next method is layering. This is a special type of vegetative propagation in which if one of the branch of the plant bends and touches the soil surface, it has a tendency to develop the adventitious roots and grow further above the soil surface. Such bent branch is known as layer, and the entire process is known as layering. If we cut this bent branch near the adventitious root, it is having the complete tendency to develop into a new plant. There are 2 types of layering. First one is mount layering and the other one is air layering. We'll start with mount layering. In case of mount layering, firstly let us talk about in which type of plant mound layering is practiced. Mound layering is practiced in those plants in which the stems are thin enough to be bent easily manually, while air layering is practiced in those plants which are broad enough, which cannot be bent. Okay. So what happens in mound layering, that we bent one of the branch such that it touches the soil surface. While doing this, the soil loosens up. We loosen up the soil and we insert this bent branch into the soil surface. After some days, we see that it develops the adventitious roots. To increase the process of rooting, we add certain rooting hormones. This soil is loosened from here. Now we will add the rooting hormones into it. We'll make a slight incision or notch near the adventitious roots, and we will add an iron bent equipment near the notch. Near the notch we will add the bent equipment. This is done so that the stem bends and reaches above the soil surface. So this notching method is done, and after notching means a slight incision is made near the adventitious root, and after that a bent iron equipment is being added so that the branch further grows upwards. It bends. For bending of the branch, this equipment is being added, and rooting hormone is also added so that the rooting process can be fastened up. This method is practiced in plants like jasmine, strawberry, apple, etc. Next one is air layering. In case of air layering, the stem is thick enough it cannot be bent manually. So in this case, see the diagram here, we'll make an incision near the node. We'll make an incision near the node, and after making the incision, we will cover it up with the moist cotton or with the moist moss. Sphagnum moss we have taken over here. After that, we will attach the tapes. We'll attach it with the help of tape or with the help of tie it with the rope so that it is fixed up there, and leave it for some period of time. In case of very hot weather, in order to maintain the moisture content, we use the water pot. We use a mudden pot over here, mud pot, in which a hole is being made and a rope is being tied up. Water comes drop by drop, and with the help of rope it moistens the incision surface. This helps in maintaining the moisture content. This method is practiced in plants such as guava, pomegranate, lychee, etc. So, in case of air layering, first method, we'll make an incision. First step, we'll make an incision. Nextly, we will cover that region of incision with the help of moist moss or moist cotton. Third step, we will tie it up with the help of tape or with the help of rope. Leave it for few days, and after few days we will see that the adventitious roots start developing from the region of incision, and this method is known as air layering. If the moisture content is reduced and the temperature is very high, so in that case we use the mud pot in which a hole is being made and a rope is being attached so that the water reaches drop by drop to the region of incision. Move on to the next method, which is grafting. It is that method in which we take the shoot region of one plant and the root region of another plant. So the shoot region taken over here is known as scion, while the root region taken is known as stalk. Later on, we will attach both of them. We will place scion and stalk one over the other such that their cambium comes in contact with each other, and we will tie it up with the help of cloth, or we can use the tape also, and leave it for some period of time. And after that we'll see both the scion and stalk have cambium have been attached. This method is known as grafting, and it is very beneficial for getting a hybrid variety, for getting a good hybrid desired variety. There are two types of grafting. First one is scion grafting and the other one is bud grafting. We start with scion grafting. In case of scion grafting, the scion can have one or more buds, while in case of bud grafting, the scion can contains only a single bud. It is a matter of just a single bud. We'll talk about scion grafting first. So in scion grafting, the scion bears one or more buds, and there are three methods of scion grafting. These are whip grafting, wedge grafting, and crown grafting. We'll start with whip grafting. In case of whip grafting, both scion, this is the scion region and this is the stalk region. We can see both of them, both the shoot region are of almost same width. the diameter of both of the shoot region are almost similar or of same diameter. Now we'll make an incision. We'll make an oblique incision in the stalk region as well as in the scion region of the another plant, such that we make such an incision in the scion region such that it fixes completely in the stalk region. After that, we'll tie it with the help of rope and leave it for some period of time, and we'll observe after few days that the cambium of both the shoot region have been attached. Now the next one is wedge grafting. Another type of scion grafting is wedge grafting. In this case, we'll make a V-shaped notch. We'll make a V-shaped notch in the scion, as you can see over here. This is the scion region of one of the plant. We have made a V-shaped notch, and then we'll make an incision in the stalk region as well, as you can see over here. And then we will fix this notch, the scion containing the notch within this stalk region, within the incision made in the stalk region, and then tie it up with the help of rope or the cloth. And after few days, we'll see that the cambium of both of them have been attached and the new plant variety has been obtained. Now the next one is crown grafting. Another type of scion grafting is crown grafting. In this case, the stalk and the scion of both of the plants are of different diameter. As you can see, the stalk, shoot region of the stalk is having a way several times more of the diameter as compared to that of scion region, while the scion region's diameter is very less. So in that case, we'll make a long wedge V-shaped incision within the stalk region. As you can see over here, we'll make a long V-shaped incision in the stalk region, and then we will make a V-shaped wedge-like incision in the scion region and fix it within the stalk region. And we will observe, we'll leave it for a few days, and we will observe that the cambium of both of them attaches and a new plant variety is obtained. So the difference between the whip, wedge and crown grafting was only with the type of incision we are making and the diameter of the scion and stalk. In case of crown grafting, the scion and stalk are of completely opposite diameters. Stalk is having a very large diameter as compared to that of scion. Scion is having small diameter. And the incision made here is again V-shaped, just like that of the wedge grafting. Next type of grafting was bud grafting. As we have seen in scion grafting, there can be 2 or more buds present in the scion region, but in case of bud grafting, we take only a single bud. Now let us see how. We have taken the bud region from one of the plant. We have taken bud region from one of the plant, and we are having a scion region. We have taken bud region from one of the plant, which will be the scion, and then we have taken the stalk region from the another plant. This is the scion and this is the stalk now. So what have we done? We have taken bud region of one of the plant, which is considered to be the scion, and we have taken the rooted region from the other plant, which will be the stalk. So this is the stalk. Okay. In the stalk region, we'll make a T-shaped incision. We'll make a T-shaped incision, and In that incision, we will insert this scion, this bud taken from the shooted region. We will insert this bud and we'll get somewhat like this. We have inserted it and later on we'll tie it up and leave it for some days. We leave it for some days and we'll see that after few days a new bud develops from the other region and a new plant variety is being obtained. This method is practiced in roses, peaches, etc. Why it is known as bud grafting? Because in this case the scion is a bud from one of the plant and we have inserted this bud in the incision made in the stalk and left it, tied it up with the help of cloth and left it for some period of time, and we have got a complete new plant in which further new bud starts developing. Another type of propagation method practiced in laboratory is tissue culture. Tissue culture is that technique in which a complete new plant can be developed from a very small part of the plant, that will be the tissue. That is why it is considered to be tissue culture. It is also known as micropropagation because a very small part of the plant, a very minute part of the plant, that is a tissue, is giving rise to the complete plant. That is why it is also known as micropropagation. So how this micropropagation is done, we'll talk about that. Firstly, we'll take the cells from the either the shoot apex region or we'll take it from the axillary bud region, and that part which we have taken, that small amount of cells which we have taken are considered to be explant. Explant means very small part of the plant. So firstly we have taken the explant and then we have put it in the nutrient medium. This nutrient medium contains various vitamins, minerals, water content, as well as plant growth regulators, artificial plant growth regulators. And this nutrient medium is taken in a conical flask, which is further covered up with the help of cotton plug so that it does not come in contact with the bacteria present in the surrounding. So it is completely isolated from the surrounding atmosphere. We have taken the explant in the nutrient medium and kept it for few days. We'll observe that a group of cells develop and it is known as callus. So this was step 1. Now in step 2, after few days, we see that this explant is now converted into a group of cell, cluster of cell known as callus. We need to keep on changing this nutrient medium as the callus is being obtaining the nutrients from the nutrient medium, and after the nutrients is being obtained it is simply plain water, and if it is not changed no further growth will occur. So we need to take care of that. The third step, we'll see that the callus further develops into embryoid. So in third step, it further grows into embryoid. And after leaving it for few days, we'll see that this embryoid now develops into a complete plantlet, small plantlet with roots and shoots. The stem has developed nodes as well. Now this plantlet is ready to be removed from the conical flask and to be grown in the natural environment. So in the method of tissue culture, firstly we have taken the small part of the plant, that is the tissue, from either the shoot apex region or the axillary bud region. We have placed it in the nutrient medium, and this part of the plant, small part of the plant taken is known as explant. After placing it in the nutrient medium, it grows into callus, and leaving it for few days, we see it will develop into embryoid, and after some days we see that the embryo develop into plantlet. This plantlet is now ready to be planted in the natural environment, in the soil, in the field. There are certain advantages and disadvantages of tissue culture. The first advantage of tissue culture is that similar characters are developed. The new individual produced will be identical to that of parent. The plants obtained will be resistant to diseases. How they'll be resistant? Because they are isolated completely from the natural environment. They are not coming in contact with any of the pest, virus, or bacteria. The plant culture is useful chemicals are obtained. We are growing certain plant varieties which are beneficial for us. Chiconin, this chemical is used in silk industry, can be obtained by this variety. By this technique, new variety of plants can be obtained. The disadvantages of tissue culture are that plants are sterile. Plants obtained by tissue culture are considered to be sterile. This technique also results in the change of structure or number of chromosomes. Now, this happens because the artificial plant growth hormones which are used in the nutrient medium results in the change in number of chromosomes. So there are certain advantages as well as disadvantages as well. Thank you.